Okay, so let's have a class, and today's class will be the review of exam 3. Now, first of all, I want to remind you, we had an extra point here. So, the slides right, the eCampus right here, which is, I put my YouTube link here, which is amino acid outside the chain review. There will be 20 points, 0.5 of name and structure. Then you go up, we also have one section, I put it there, is aerobic respiration ATP calculations. So glycolysis and TCA cycle, structure, 1 point, name, 0.5 points. And the whole thing, we use the old calculations, 38 ATPs. If you want to use the new one, that's okay. But you have to keep consistent, okay? So that's, and I said the calculation is required. However, the structure and the name will be extra points. And so there's a lot of points in. So let's talk about our practice questions. And we'll do the review. If the knowledge which is related to the certain slides, we're going to talk about that. First of all, do not worry about all these red one. The red one I didn't talk this semester, so we can keep it that. Okay, don't worry about these red one. Some questions I missed, also jump it. Okay, let's see. First question first. A non-promoter, a non-protein component of an enzyme that is loosely attached to the protein component is referred as what? If it's loosely attached, then what? It's a coenzyme, is that right? So where it is, if you forget about this, you need to go back to see the slides. The slides will be right here. That's a time for you to use these review questions to study. And I want to also tell you, if you know that our practice exam and the real exam are highly correlated. So still some people tell me they don't want to do it because they don't believe it. Of course, it is correlated. Look at this. Coenzyme loosely attached can act as carriers and shuttles. Okay, so now, of course, another one, prosthetic group is firmly attached. Okay, so here's the thing. Coenzyme, loosely attached. Okay, prosthetic group is firmly, or we say closely attached. Okay, so that's the first question. This is an example for you to study. The law of thermodynamics states that physical and chemical processes occur in such a way that randomness, which means disorder, increase to a maximum. Of course, second law, is that right? It's so simple. And we said the first law is energy never generate, never disappear, only transfer. So, what is the first law? The thermodynamic law. Basically, energy transfer from one form to the other form never disappear. What is the second law? And so simple is disorder increase. Is that right? If you can do the review. Okay, this is a question a little bit difficult. Below are the components used by a cell bacillus. When you see cell bacillus, it's just a bacillus. To make energy from oxygen and iron. So we have a cytochrome A, cytochrome C, ferric chloride, ferric oxygen, rust cyanine. Please put these components in the correct order in the membrane and show electron-proton flow with arrows. What you should do? This is, we go back to see our slides, still right here. Which question it is? Right here. On the top is more negative, which means more smaller. More small number indicates it's a good electron donor, and at the bottom is more positive, which means it's a better electron acceptor. Then, when you look at the question, what's going to be the order then? Which one will be at the bottom? Oxygen and water, is that right? Why? 0.82. Which one will be on the top? Ferric. Okay, then go here. This is 0.77. What's the second one then? Rustocyanine. 0.78. Then what's the next one? Cytochrome C. 0.79. What's the next one? Cytochrome A, is that right? 0.80. That will be the electron transfer. Why will be like this? Because more positive is more electron acceptor, and more negative is an electron donor. Now here we don't have a negative, but smaller, which means more negative. So electron donor. Okay, so that's a question. You need to put this way. Okay, if you only put in order, we lose some points. Okay. This question, which means it's a little bit difficult because it's an application of this one. Okay, you need to know. So when you study these slides, you should all put them together. These three slides, lecture 1, slides 15, 16, and 17, you should all study them together. That's what's the purpose of these practice questions. Next one. Which of the following process generates the greatest amount of energy? Fermentation, substrate level phosphorylation, anabolism, glycolysis, anaerobic respiration. A, B, C, D, E. Don't let you guess too much. Where is going to be the answer? Okay, let's go see. Slides number 2. And I said this chapter, the very important, is this table. How many aerobic respiration generate? We already said 38. How many fermentation generate? Only 2. How many anaerobic respiration generate? 36 or less. So, what is the answer here? Anaerobic respiration. Glycolysis is only 2. Fermentation only 2. Anabolism is not generate ATP. Anabolism is using ATP. Is that right? Substrate level phosphorylation, that is not a process. We talk about substrate level phosphorylation. What is that? If you want to review, go back to study. Substrate level phosphorylation is all these slides right here. A typical one is this one, and this guy. That is substrate level phosphorylation. What we talk about is ATP, is ADP, then becomes ATP. Okay, so ATP itself can do the phosphate transferring, and what is the transferring power is about 45.6. So anything, the power more than that could do substrate level phosphorylation because the delta G is more negative. Anything less than that, you can't do it. Although it looks like they carry phosphates because their phosphate transfer power is less than ATP itself. So we mentioned about in the lecture. So this is the slides talk about substrate level phosphorylation. It does not really mean how many ATP you're going to generate. It's just a concept. And the contrast concept to that is oxidative phosphorylation. So then you go ahead to study what is oxidative phosphorylation. Is that right? That's what you're going to study. What is oxidative phosphorylation? Right here. Peter Mitchell, the chemosmosis. So that's the way you're going to study. Okay? Sulfur anilamides or antibiotics, that is function by inhibit bacterial growth by binding to the active sites of the enzyme in the TCA cycle. That is A. Inhibit the enzyme by binding to allosteric sites. Inhibit the synthesis by vitamin committed normal, blah, blah, blah. Which one is easy? This is a question, easy question, is that right? Pick the longest one, C. But that's a trick for you how to do the question, then how you got to understand it. Then you go back to C. That's what I'm showing you how to do these questions. Then you go back to C here. Go back. Right here. That is competitive inhibition. So when you study, mark these slides is important. Can you see the sulfur drug and the PABA? They look exactly the same. PABA is a precursor to manufacturing folic acids because it looks very similar. So instead of PABA with enzyme, the sulfur drug will be there. Okay, so it's a competitive inhibition. Then go ahead to study immediately. What is another way? Is non-competitive allosteric regulation. What is a good example? Lactose operin, is that right? Allolactose is an inducer. That's an example of allosteric regulation. Okay, so that's two slides, you can combine them together. So the answer is C. Very obvious. Question 7, A, B, C, D, E, F, G. That's okay. It's just practice. They won't have A, B, C, D, E, F, G in the real exam. Okay, only is 4. The transfer of electrons and hydrogen by a dehydrogenase to an organic molecule as the terminal electron acceptor is called what? Respiration, anaerobic respiration, substrate level phosphorylation, fermentation, glycolysis, end product repression. Some of you don't even heard about. So let's talk about electron donor and electron acceptor. You pretty much forgot, but we have to refresh you. If electron donor is an organic chemical, and electron acceptor also is an organic chemical, what is this one called? Fermentation. Is that right? So very simple. Pyruvate going to lactic acids, and the lactic acid we call it the derivatives of pyruvate. If electron donor is an organic chemical, if an electron acceptor is an inorganic chemical, what we call it? Respiration. Then respiration have two different categories. If this inorganic chemical is oxygen, we call it aerobic respiration. If it's other than oxygen, then we call it anaerobic respiration. And again, how many ATP generated? 38 ATPs. If it's complete, this is between 2 to 36 ATPs, depends on the components. Remember? We have that picture. So what is the answer here? What is it here? Fermentation is D. Okay, then go ahead to review this. Which of the following is the best definition of fermentation? Right there is the best definition. So what it is? The oxidation of a carbohydrate with organic molecules serving as a final electron acceptor. The answer is C. This is another trick. It's too long. You look at that. So you say these are difficult questions? Not really. You can't even guess. I believe there's only 30 seconds left. I'm not doing strict timing. If there's only 30 seconds left, you will just pick C. Is that right? And C usually you will pick because right in the middle. So it's not really a difficult question or looks like difficult. Which of the following, next one, which of the following is used as an electron carrier by living organisms? Okay, then go ahead to see the slides. That's why I tell you all these things, it's all related to the slides. It's not really I made up something. Look at here. That's the electron carriers right there. NAD, NADP, FAD. Is that right? So that's the electron carriers. So what's the answer? All of the choices. When the end product of a pathway inhibits catalysis of the first step of that pathway, this phenomenon is called what? A, B, C, D. A, B, C, D, okay? Which one? What are we talking about here? Is that end product could be effects intermediate and substrate A? So feedback inhibition. Okay? Which is a good example for that? It's a tryptophan. Tryptophan operon is a good example. We didn't talk about too much, but you should know that's a feedback inhibition. So click here, connect to that slides. Then that you can study. Okay? Don't worry about that 11. This question, I hope everybody can do. Pyruvate go to lactic acid. NADH become NAD plus. What is the name of this one? Fermentation, is that right? What is this called? We should call this as hormone fermentation, because it's only the final product is lactic acid. That's the first question. Second, please name the reduced products, oxidizing product, reducing agent, oxidizing agent. Tell me again, what is reducing agent? What is reduced product? Oxidize the product. Reducing agent. Okay, oxidizing agent. This we can talk a lot, but I will give you some trick to learn, okay? What is a reduced product? Lactic acid, is that right? So what is oxidized product? That's a well, it's lose electron. What is oxidized or reduced agent? NADH. What is oxidized agent? Pyruvate, is that right? Okay, if you don't know anything, I'm just guess. You at least get 1 point. You write and everything is the same. Is that right? Or another way, write 2 NAD plus 2 NADH. One way or the other, you get 2. These questions are really nice. It's although difficult. Don't left it empty. Couple of years ago, that's an exam question. Blank, 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 blank. Somebody left it empty. I said, why? You just write something there. There are only 4 chemicals. Write 2 of them. You want gambling? 2, 2, 2, 2. You get 2. You write everything is the same. You get at least 1. Better than you left them empty. Is that right? So something you need to learn when you do a test when you don't know how to do it. Okay? So that's one. What is the biological function of NAD in this reaction? There's a well there, but carrying electron. Electron carrier. So that's very simple. Electron carrier. Name one product processed by this reaction, name the microorganism related to that. Did you eat yogurts, smoothie? So what's that? Dairy products, yogurt. What's the name of the microorganism? Lactic acid bacteria or lactobacillus. That's so not difficult, the lactic acid bacteria. If you don't know lactobacillus, that's okay. Yogurt. Is that right? That's not a really difficult question, but believe me, some people are still going to missing this. This is a very typical example for understanding, reducing oxidized reaction, understanding fermentation. A very simple question, but tests you all the knowledge about that. 13, don't worry about that. Which of the following can be used as an electron acceptor during anaerobic respiration? we just said. Right there. So this is the key. I'll tell you one thing. This is a golden rule for rules for exam 3. If you understand this or lots of questions, you can do it. This is the golden rules. So what is that? All of the choices could be. Okay. 15. Which of the following is not true regarding ATP synthesis? And this is a little bit difficult because we didn't mention this too much. They require proton motive force to make ATP. That's correct. They span the inner membrane of the mitochondria. Yes, that's correct. The proton flow is outward during ATP synthesis. That is not correct. The proton flow is backwards. This is a question a little bit difficult, to be honest, because it's come back. Look at this. So what is chemosmosis? During the electron transport chain, rely on dehydrogenase. What is dehydrogenase? Take out the hydrogen out. Then they have a proton motive force. Proton is H+. They accumulated, it's a potential energy. Then generated become a transferring become kinetics energy, activate ATP synthesis. If it's H is too much there, proton too much there. Then come back, coupled with active transport of nutrition and flagellar rotation. That's a chemosmosis. Peter Mitchell in 1932 mentioned about that. And later on, it's a Nobel Prize. So that's something we want to talk. So it's a C, okay, not outwards, it's inwards. The hypothesis that proton motive force drives ATP synthesis is called chemosmosis, right on the top. When you read these slides, can you please read these titles first? Okay, that's the first thing first. So make sure you understand and read these titles first. Organisms that form lactate, ethanol, and carbon dioxide as end products of glucose fermentation are called, this is called a heterofermenter. Remember, this is a homofermenter right here, heterofermenter. Which means you generate other products like carbon dioxide, alcohol, or others, maybe aldehyde, all those things, okay, other than lactic acid. Basic requirements in this classroom. Can you do the calculation? I don't need to repeat it one more time. Is that right? Look at the video. Can you do it? We don't need to talking anything about the structure. You need to know how to do ATP generation. If you still don't know, please go ahead to study these slides. If you still don't know, or right here. Either way are fine. Here is what are my requirements. Okay, when you do the calculation, make sure at the top of your answer, very clear. NAD reduced to NADH2 is 3 ATPs. FAD reduced to FADH2 is generate 2 ATPs, or 2.5 and 1.5, but not mixed. I would suggest you 3 and 2. It's easy to calculate. Make sure at the top, right there. That gave you some points. I gave you 2 points if you write it. Okay, so there is no way this class you end up with C. At least you're going to get a B. So there's all these questions. Well, if you write these, you left empty, I think you understand, I'll give you 2 points. Okay? So make sure people understand how you calculate it. You can do 2.5, 1.5, but not mixing. Okay, so this is 2 slides is important. We talk about the calculation. And this is the chemicals right there. Okay? All right there. But you can look at my video, is more clear. Oh, this is tough. A, B, C, D, E, F, G, H, I, J, K. Don't let you guess. If yeast metabolized one molecule of glucose to acetyl alcohol in the absence of oxygen, how many net molecules of ATP would be obtained? That is 2. Because it's a fermentation. Fermentation only generates 2 ATPs. Okay. 20. How are aerobic respiration and anaerobic respiration similar? I told you this is the golden rule. Right there. What's aerobic and anaerobic respiration similar? Both utilize inorganic molecule as a terminal electron acceptor. So the answer is C. 21. Probionic bacteria makes probionic acid, acidic acid and carbon dioxide in Swiss cheese. This product is result from fermentation. That's not difficult. Where it is the answer. This, you need to go back to see that slides. Okay, this is a product. Probionic bacteria is the first one. Generally, is probionic acid and Swiss cheese. Swiss cheese, you see the whole there is carbon dioxide. So that's the answer is right here. Okay. Okay. Citric acid cycle. Just want to tell you, it won't be question like that. Because if the question like this, you don't need to write. Is that right? You just copy. So it will. This question probably not going to test you, but we can do a review. Are there oxidative phosphorylation in this cycle? Yes. Any places when you have NAD reduced to NADH2 is oxidative phosphorylation. This is, we have to make it, you understand, and we don't talk about chemicals. This question, we don't really talk about chemicals, but you need to know pyruvate become acyl-CoA. Okay, the circle. So what happened? Right here, NAD reduced to NADH2. Right here, NAD reduced to NADH2. Then here is GDP, becomes GTP. Here, FAD reduced to FADH2. And right here, NAD reduced to NADH2. And don't forget, transition step NAD plus reduced to NADH2. So, is there oxidative phosphorylation happened? Yes, of course. All these are circled. Whenever you see NAD reduced to NADH2 or FAD reduced to FADH2 is oxidative phosphorylation. Phosphorylation, you can just write a P. Okay, it's easy. Are there substrate level phosphorylation? Right there. Label the place where so-called transition step, right there. Or define the precursor, metabolic precursor, name one chemical in this cycle related to the biosynthesis of protein and fatty acids. This one I didn't mention this semester. But if you want to learn, you could know it right here. Glycolysis, if you want to see it. Alpha ketoglutarate is a metabolic precursor. It's related to protein and fatty acids. Okay, alpha ketoglutarate, you know about that. What is metabolic precursor? And I didn't mention that concept, so don't worry about it. I used to talk about that. Next one. In aerobic conditions, it takes sugar to produce the same amount of ATP when compared to the anaerobic conditions. More or less. Less, is that right? Yeah. Which of the following can be used as an electron except during anaerobic respiration? All of the choices. We already mentioned. List the central metabolic pathway. I talked about that before. It is fermentation, glycolysis, and the citric acid cycle. But we don't teach this semester. Distinguish between first, second law, dynamic law. We have slides for that. This is an easy question. So go back, when you see this question, go back to study right here. What's the difference? Right there, first law, second law. Who proposed the chemosmosis theory? Peter Mitchell. Name one final product generated from pentose phosphate pathway. What is that? NADPH. Is that right? Pentose pathway is right here. I didn't talk about this one too much. Right there, NADPH. Pentose pathway, because it generates precursor material for synthesis, DNA and RNA. This one I didn't talk. Okay, that's down, the first one. Second one is a little bit tricky, because it's talk about the genetics. Let's see, is this a tricky one or another tricky one? Okay, this is maybe the easy one. Let's talk about the difficult question first. Shown below is the nucleotide sequence of a fragment of double-stranded DNA from E. coli. The start site of the transcription is indicated by the locus of A. Okay? Where is the start one? Where is the locus of A? Did we see it? Right here. It's the locus of A. On the sequence above, show on the label where the promoter is located, related to the start site of the transcription. This one is a little bit difficult. This is another golden rule. These are golden rules here, okay? Five prime, 3 prime, 5 prime, 5 prime, 3 prime. Plus 1. TA, AT. 70 BP, TA, GA, CA. Is that right? TTGACA, sorry. That is called minus 10 BP. This is minus 35. This area, the whole area, per molar, combined with RNA polymerase. This is the golden slides. So we're going to go back to look at this slides number 3. Okay, this is the golden slides, what I talk about. I said it's very important. Right there. 3 star, mark it. What is the promoter? Minus 10 sequence, minus 35. Minus 10 sequence, we also call it a promoter box. The whole area is promoter combined with RNA polymerase. So how are you going to label here? Where's the promoter? TA, AT. This big chunk, sorry, I can do it. Highlighted like this. Then, let me try. Sometimes it doesn't work. Go back. Like this. Okay, good. Okay, same thing. You go down, you have to label. Okay, you know that. Okay, we're gonna do it. Just for somebody doesn't understand. Go here. Okay, then we go also go here. That's a promoter. Okay, show the promoter. Name the enzymes that recognize the promoter sequence. I already said RNA polymerase. Right there. Name the protein subunits that allows the enzyme you identify in question 40 to recognize the binding and the promoter sequence. RNA polymerase, which one to help recognize the promoter? Sigma 70 factor. Remember, alpha 2, beta prime, and omega it's a coenzyme. Sigma factor is a coenzyme, so sigma factor. Write out the mRNA transcript and be sure to label the 5' and the 3' ends. On the mRNA transcript, circle the second translated codon indicate which amino acid is coded by the codon. Okay, here is the thing. Which one is to start? What is AGG? Shai-Dagano sequence, is that right? Recognized by 16S RNA. What is the stop code? Is AUG, so you need to find is ATG right here. Can you see it? Okay, then what is the second one? AUC, is that right? What is the code of AUC? I think it is isoleucine, probably. Don't worry, we won't test you. If you need it, I'll give you the code. Genetic code. Okay, let me see it. I think it's isoleucine, if I remember it. I don't remember everything. Sometimes I remember some, so I'll go take a look. Let's take a look. Is that isoleucine, AUC? Yeah, it's isoleucine, right there. Okay? So this is a question. This question is a little bit difficult, to be honest. You can do all of them. Circle the second translated codon. You need to know this is AGG. Find the AUG first. The second one is AUC. And also, don't forget, this is 5 prime, and this is 3 prime. You can write it, is that right? It's all the same. T becomes U. So if you write, if you want to write, lots of the years there are some people writing, write the whole thing. Of course not. You start right here. So what are they? What you should write? If you very want to write, let's go here. Then A U G. So then A U C. So you have G G G. So you have C G A. So you have G A C. And that is 3 prime. Oh, sorry, this is too large. That is 3 prime. See? Don't forget, 5 prime to 3 prime, 3 prime to 5 prime. So it's 5 prime to 3 prime. Okay? And this is coding band, and this is template bands. The only difference is the real mRNA is U replaced with T. So the second one is right here. Is that right? So that's isoleucine. Okay. Question number 4. This one you don't have to do everything. Well, we just something I didn't mention this semester. Shown below is a partial sequence of an mRNA molecule. Could this RNA transcript be an mRNA? Why or why not? What's the significance of the underline? Can this be transcribed to mRNA? There's a Shai-Dagano sequence right there. And they also have an AUG right there. So, yes, it could be. Because Shai-Dagano sequence and the initial code are right there. Okay. Would the deletion of the base marked G1 have any sequences? Explain your answer. Yes, because when you deleted it, the AUG will be shifted. The initial coder is gone. Okay. What would be the consequence of deleting the lowercase, bolded based on the sequence above? The best answer is it could be or could not. It depends on the reading frame at the back. Because it could be, you made a mistake here, but later on you fixed it. Okay? And also it depends on the mutation. If mutation happened on a third box, it's called a wobble, it doesn't matter. Don't worry about the 45 and the 44, because we didn't talk about this semester. It's a mutation, so we don't test you. Okay, these two you don't have to worry. So these are some questions we mentioned. These two really study well because it's difficult. Let's go over all these. 2 strands of DNA molecule are, that is oriented in the opposite direction. What are the anti-parallel? Is that right? A consensus sequence in the untranslated leader sequence of prokaryotes mRNA, which serve as the ribosome binding site, is called what's the sequence? Is untranslated leader sequence. It's stop codon, is that right? Start codon, is that right? It's called it's called what? A consensus sequence in the untranslated leader sequence of prokaryotes serves as the ribosome binding site is called what? SD sequence. Is that right? AGG. Okay. Amino acid tRNA synthesis catalyzes covalent addition of FMET, formyl MET to tRNA. MET. Then MH. Then is that right? That's what we said, the formula. Okay. Methionine. The genetic information in DNA is divided into units called what? Genes or genotype? It's genes, is that right? Okay. The basic difference between DNA and RNA rather in the sugar and the pyrimidine bases. RNA has ribose and uracil, whereas DNA has deoxyribonucleic and thymine. Yes, it's correct. So the answer is true. Which of the following is not a pyrimidine? Which one is not a pyrimidine? Adenine is what? Is purine, is that right? Cytosine is what? Is that purine? Say which one is not a pyrimidine? C or D? Which one is not a pyramid? Yes, uracil. Okay. Adenine is what? Is a purine, is that right? Then which one is not a pyrimidine then? Yeah, the answer is A. Yeah. It's a very easy question, but lots of time people doesn't get it. People just confused about it. Okay, A and T, G and C, AG are purine, CT are pyrimidine. U is also a pyrimidine. Is that right? So A is not. Okay. Which of the following is not a complementary base pair usually found in the DNA molecule? This is easy. Which one? A and C is found in RNA. Okay. Which of the following is true about structure of DNA? Purine pyrimidine attached to the one carbon. Okay, that's correct. Yeah, we draw the structure one carbon of the oxyribose. Purine and pyrimidine is joined by phosphodiester bonds. Is that because they joined by phosphodiester bonds? Purine and the pyrimidine. Number 3 is correct. Adjacent bases attached to each other is base 0.34. Which one is true about it? Everything, is that right? Yeah, all the choices. Is that correct? Yeah, all the choices. Which of the following nitrogen base is usually found in RNA and not in DNA? That uracil. Then what is the replication? Semi-conservative. Okay, C. Y-shape part of the DNA molecule where the actual replication process is called a replication fork. It's always happen at a replication fork. During replication, the two strands of DNA molecule unwound from one another by enzyme is called helicase. Is that right? There's no unwindedness. Don't worry about that. That's just silly choice. Helicase. The coding sequence in the DNA of is normally continuous. That is not interrupted by non-coding sequence. What is that? What is the difference between these? Plants, mammals, and the most insects is eukaryotes. That's different from prokaryotes, so the answer is A. This is, I didn't mention this semester. You know the transcription, translation are coupled for prokaryotes, but for eukaryotes it's not, because you have an exon and an intron. And the intron have to cut, then the exon will be connected by the enzyme called splicism. Is that right? So, but you should know this is the answer is A, because B, C, D are the same. The region at the RNA polymerase binding is a promoter. That's B. Unexpressed region of split genes are called, what is that? Intron, is that right? Unexpressed. The pyramidal box centered approximately 10 BP base pair upstream the start site of the transcription. So that's C. The genetic code is to be because more than one codon will specify a particular amino acid. What we call it? Degenerate. Okay, that's what we call it, a degenerate. Which is not a termination codon. AUG is not, it's a start code. Molecular deliver amino acids to ribosome during translation, tRNA, transferring RNA, carrying amino acids, but make sure this is anticodon. So what's the tRNA brain? Anticodon, okay? Ribosome can attach to the mRNA and begin translation even though transcription has not been completed. Prokaryotes, this is the same thing. B, C, D are the same, it's all eukaryotes. The reaction sequentially link the adjacent amino acids in growing polypeptides. It's called transpeptidation. Okay, so that's B. What's the function of DNA polymerase 1 and 3? What's the function of DNA polymerase 1? Cut RNA primer of the Okazaki segments, is that right? And the DNA ligase will have to connect to them. What is the function of DNA polymerase 3? Polymerization, the second one. Proofreading. That's a fidelity issue. So DNA polymerase 3 is proofreading and polymerization. What is Okazaki segments? It's in the lagging band. There's a small DNA sequence leaded by the RNA primer. Is that right? It's complementary to the one strand, the lagging band of the DNA, and they have a small segment because you can do the replication from the lagging band directly. So that's Okazaki.